I just wanted to welcome everybody to the New York Invasive Species Awareness Week Invasive Species Spotlight webinar. Today we're going to be talking about species of concern all across New York State. My name is Brent Boscarino, and I'm the Invasive Species Citizen Science Coordinator with the New York, New Jersey Trail Conference and the Lower Hudson Partnership for Regional Invasive Species Management. We'll be talking about those terms in a second. So first of all, just to give you an update, I know some of you on the call today, but not all. My role at the Trail Conference is to essentially try to inspire action. So to get volunteers like yourselves and community members who are interested in this kind of stuff, who just like to be outdoors for some reason, to get outdoors, explore, to learn about nature, and then to give back, somehow protecting our natural areas that we love to visit and are a part of our lives. So I do that by helping to coordinate the citizen science or community science powered invasive strike force. And what that is a program that we launched in 2011 as an effort to essentially detect and then later manage the spread of invasive species in a lot of the forested parklands. I'm based in the Lower Hudson PRISM region, so Lower Hudson Valley of New York State. And I work with all volunteers of all ages to essentially track invasive species and protect our native habitats in the Lower Hudson Valley. And because we're hosted by the New York, New Jersey Trail Conference in Northern New Jersey as well. So in that big introduction there, I used the word PRISM, and we're all here, and why I get to do the really rewarding work that I do. And it's because of this PRISM network. And that stands for Partnership for Regional Invasive Species Management. And there's 8 different types of these cooperative partnerships throughout New York State. So I happen to be located within the Lower Hudson PRISM region, but I know that there's people on this call across New York State in different PRISM regions. Each of the PRISMs themselves are hosted by a certain nonprofit. So we are hosted by the New York, New Jersey Trail Conference, but all of our work, regardless of whether you're out in Western New York or through NYSAW, which is in Long Island, where all of our work is contracted through New York State DEC via the Environmental Protection Fund. And I feel so blessed to be able to do this work and work with amazing volunteers like yourselves. Just as a general overview of what we do as a PRISM. Well, every PRISM region has a slightly different focus, but in general, each of the PRISMs are going to be involved with some type of removal and control work with invasive species. So in our PRISM, we work in both aquatics, so in lake systems, we work a lot with the Hudson River, but we also do a lot of work in our forests and protecting that. We have seasonal crews. They just started a couple of weeks ago and are doing an amazing job, and that's run through AmeriCorps here in our Lower Hudson PRISM region. A lot of what we do is surveying and mapping too. So across the PRISMs, I know that there's a lot of great survey and mapping work that's done that helps drive management decisions involving invasive species. But really where a lot of the planning happens is within the PRISM network. So remember that a PRISM really isn't an entity upon itself. It really relies on community members like you and local organizations that have a vested interest in invasive species that do the prioritizing and coordinating. So that's usually done through meetings and sharing of information, working with a great network of environmental groups and concerned community members like yourselves. We also, across the PRISMs, not just in our PRISM region, do a lot of teaching and informing. So whether that's working with school groups, going into schools, or running official training workshops. A lot of the PRISMs, not just ours, but again, across of New York State, have watercraft inspection stewards, and they do a lot of boater outreach involving aquatic hitchhikers and aquatic invasive species. So this is what the PRISMs generally have in common. And I will say that one of the main focuses of what the PRISM network does and what the staff and the volunteers and what makes us go is the idea of early detection and rapid response. So that we call that EDRR. And essentially what that means is that people like you, members of the public, our staff, we are really, really focused on getting the word out to spot these invasive species, what right in the initial parts of when they come into our area, because it's that at that critical time when the population hasn't had time to build up to these numbers where it's very, very difficult to control, that management success can be really high. So we have what was called a window of opportunity. And it's right at the beginning of when species start to come into a region that management success can be high. We can put in really good high priority management strategies to hopefully like eradicate the species, or at least make a major dent so that it's not really influencing our native habitats that we treasure and want to preserve, right? Because typically if you let time go by and when invasive species really take a hold, that's when a lot of the resources of money and time and energy spent to kind of beat it back gets a little overwhelming. So a lot of the species we're going to be talking about today fall within this early detection and rapid response window. And I feel blessed to work with amazing colleagues across the state. And we asked each education and outreach staff member across all the PRISMs, and again, you can see all the different regions here, to nominate one featured species of concern in each of those regions. So we're going to be talking about 8 different species today. And these are species that the E&O staff members said that they're either actively focusing on through management or particularly early detection in their region. Maybe the species that they decided to nominate for this webinar today is unique in some way or a focal point for that PRISM in terms of management or early detection. As I said, we've got a good mixture of aquatic and terrestrial species and plants and animals. So you're going to be learning a lot. There's like nice range of what we're going to be talking about today. And we'll be focusing primarily on ID characteristics so that you can come away from this webinar and go out for a walk or go visit your favorite lake and have a picnic, be walking around afterwards and be like, hey, you know what? Brent told me about that. And I know that's a species I should be on the lookout for. And now I know to report so we can do something about it in terms of rapid response. So these are the 8 species we're going to be talking about today. I won't go over them like right now and read them off in a big list. You can see them in front of you. But we're going to be making our way west to east, kind of in a clockwise direction. We'll start with Western New York and then make our way down and across through Capitol Mohawk to Lower Hudson and then into Long Island PRISM. And these are the species we're going to be focusing on and kind of making our way through. So now again is the time to kind of get your thinking hats on and sit back and kind of learn about some of the ID characteristics of these species of concern, what PRISM region is really focused on that species, and start putting this model of early detection and rapid response into place so that we can start making a difference in our local ecosystems. So the first question is, what are the ID clues do we look for in each of these 8 species that we're going to be featuring today? And we'll roll it out. The first one, the first nominated species from Western New York PRISM is mile a minute. And those of you who work with me on my job here in Lower Hudson PRISM in the Invasive Strike Force program will recognize this one. This is one we have here as well as Western New York. It's called mile a minute. And mile a minute, as you can see here, is an herbaceous or kind of like fleshy trailing vine. It creates thick blankets over other vegetation. So if you look at it here, when you think about vines, you tend to think of them as like growing straight up trees, but mile a minute really tends to blanket shrubs and understory, and it kind of grows out horizontally, right? And it's known, as you'll see in this field ID video that I'll show you in a second, it's really known for its highly distinctive equilateral triangle shape. It's unlike anything else you're going to see out there. And why is it called mile a minute? Well, it grows 6 inches per day, up to 30 feet in a year. It's very hard to get rid of. Its seeds are dispersed by water and contaminated soil. It can hitchhike on your clothes. We'll find mechanisms for grabbing onto you, for moving. But it is an annual, meaning that the plant will die back at the end of the year. So only the seeds are going to survive and not the roots. But those seeds can survive and persist in the soil for up to 7 years. So with that, I'm going to show you an ID video that you can use and think about the next time that you go out and want to look for it. So here we go. This is mile a minute and what it looked like just a few weeks ago from in the lower Hudson PRISM region. Again, we have it here as well as in Western New York. Here's a look at the beginning stages of mile a minute vine in this forest understory region here. And there's a couple of things that make mile a minute stand out from other forest understory species. So first of all, take a look at its leaf shape. It's almost unlike anything else in this area. It's like almost exactly an equilateral triangle. Typically lime green in color. You can see that there is a garlic mustard leaf right next to it that kind of has. So garlic mustard is another invasive species that I'm sure that you have. It's prevalent all throughout New York State. And you can see it here. It's got more of like a flowery feel to it. So this is called a garlic mustard rosette. You can see it's got big teeth or notches towards the end. Mile a minute is just a smooth edge to it and just a triangular shape. It's more of a flowery tooth edges to it. I mean, there's none of that happening here. Just almost pure lime green equilateral triangle. You'll also notice that it's not just those triangular leaves, but look right next to my thumb. There is a little saucer-shaped leaves at each of the nodes where, you know, each of the stems are joining together, almost like you can put a little teacup on it or something. So that is, you know, those saucer-shaped leaves are another hallmark of mile a minute. And even though this is early growth in the year, in May, you can start to see the barbs, the little spikes that are coming off of the stem of this. Again, like little. And you can see where I'm talking about here. It's got those like little dorsal. Oh no, what happened there? These little. It's got the little dorsal fins of that. What we jumped ahead here. I don't know what happened. I shouldn't have been clicking around. Let's get back to it. So that is, you know, those saucer-shaped leaves are another hallmark of mile a minute. And even though this is early growth in the year, in May, you can start to see the barbs, the little spikes that are coming off of the stem of this. Again, like little tiny shark teeth or shark dorsal fins or something. They're coming out right there, guys. Recurved barbs and spikes that allow it to grab onto things and sort of, you know, have that like vine you know, adherence to whatever it's trying to grow on. And again, this is early stage of growth here in this forest understory. And eventually, you know, this will just keep growing mile a minute. That's why it's called it, and eventually blanket all of the leaf litter. So that's mile a minute for you. It's very, very distinctive lime green color to it. And you know, it can't be confused with all that many things. I'll show you one lookalike. So again, just a quick review of what we just learned there. Triangular leaves. There's little shark tooth barbs on it that it's using to take purchase and kind of make its way across horizontally across shrubs and forest understory, the mint green color. And again, the stems have these like really kind of, they're actually pretty cool round leaf-like stipules, they're called, like a little leaf-like appendage that you could put a little teacup on. The other thing to be on the lookout for is that they have these really interesting blue berries that ripen in the late summer. And if you've ever been walking by it, it's pretty obvious, and they'll persist out throughout most of the fall. And you might see those ripening in the late summer if you have mile a minute vine in your PRISM region. And as I said before, the only thing that a lot of people may confuse it with is a type of bindweed. However, bindweeds do not have those prickles and those like barbs along its stems. It also doesn't have that saucer shape like leaf stipule on it, or that little like thing that looks like a leaf on it. And it's just it's just not quite that equilateral lime green equilateral triangle. There's more like bindweeds, a little more heart shaped than that. All right, in terms of mile a minute management tips, if there's no berries present on mile a minute, we recommend that you pull out the plants or roots and you can leave it in the sun with the root exposed, or you can bag it up and just monitor the site and repeat as necessary. So that if this is in your backyard, this is something that you can do. If the berries are present, we recommend that you pull out everything, put it in a bag, and then dispose those bags. Do not compost it, right? So if you can pull it out of wherever you're pulling, try not to compost that. And again, the berries are how it's reproducing those seeds, right? So got to make sure that it gets removed from the area. If you can't haul it out in your middle of nowhere and you're pulling out mile a minute, you can leave those bags out in the sun, right, for like 4 weeks, and that should kill the seed viability as well. So That's kind of a last resort type of thing. The other thing I wanted to point out is in the Lower Hudson Prism, we have a mile a minute project, and this has been running for a number of years. It's not running this year, but there is a little stem boring weevil that only feeds on mile a minute. And so these weevils were released strategically in our region, and it helps to defoliate and stress out the mile a minute plants. It decreases seed quantity and quality. And again, it feeds exclusively on mile a minute. And you can see just how small it is. There's a penny up there for reference. So these are really tiny little things. So if you're out and you see these little bullet holes in mile a minute, you know that that weevil's doing its job. So just be on the lookout for that. It also feeds on the nodes of mile a minute, and it's done an amazing job to kind of curtail it, at least in the Lower Hudson Prism region. All right, we are on to our next focal species, and this one comes from the Finger Lakes Prism. And I know we have somebody on the call from that region today, and I'm sure that you have heard about Hydrilla. So Hydrilla is found in Cayuga Lake in the Finger Lakes, and I know that there's a massive control problem or a control program that's going on there, and I'll mention that in a little bit. But Hydrilla is an aquatic plant that's native to Korea. It was introduced to the US in the 1960s and became one of the most popular aquarium plants. So again, like, you know, just people kind of bringing them into their aquarium, then disposing of them in natural systems. And that's how it kind of continued to spread. We also have it here in the Croton River in the Lower Hudson Prism region. So there are a couple of different areas in which it's found. So it's found in over 30 different states. It's a huge problem in Florida in particular, but there are 12 counties in New York State with reported populations of Hydrilla. It is now prohibited in terms of both sale and possession in New York State. So we're really hoping to get to this before, you know, it spreads even farther. And I'll show you what some of the management has been up to this point in its invasion history. In terms of its identification, so this is a good look at Hydrilla, and it's got these really cool what are called whorls. So you see these leaves here, and how they're sort of like whirling around that stem there. Those whorls usually come in 4 to 8 or usually 5 blades that are whirling around that stem. So interestingly, you know, just one of these whorls or these nodes, it is left behind, a new Hydrilla plant can grow from it. It can reproduce through fragmentation. So you got to be really super careful if you're trying to remove this. Those leaves also have a toothed or serrated margin at the end. So if you looked at the edges of this leaf, it would have like little edges like teeth on the ends of it. Another way to identify Hydrilla, and this is from the New Croton Reservoir in our area, are through what are called tubers. And don't they look like little potatoes, because potatoes are tubers too, then they're small white to yellow potato-like tubers attached to the roots. So again, what a tuber is they are swollen portions of an underground stem or rhizomes, and they store nutrients for plants to use during dormancy. And they're eaten by waterfowl, and they still remain viable. And little Hydrilla can also shoot out of these tubers and these little potato-like endings on this Hydrilla, and provide new shoots and new Hydrilla from that, as well as another mechanism for spread. And then the third primary mechanism for spread is through these things called turions. And turions are really key to Hydrilla's reproductive success. They're specialized overwintering buds that can detach from the parent plant. They remain dormant usually until spring, but this is how they will survive even like really environmentally stressful conditions. So they'll remain in the sediments and stay dormant until conditions are right to sprout and grow. Usually that's in the springtime, but if there's some sort of like environmental disturbance, they can linger in the sediments for up to 4 years. So lots of different ways and reasons why Hydrilla is really hard to get rid of, right? Fragmentation, turions, and tubers, all mechanisms for reproduction. They form these dense mats. So again, they're kind of these, you can see it in this picture here in the Croton River, form these dense mats that are like smothering out light and nutrients. They outcompete our native plants. Anytime you see this type of density in an aquatic system, you know that they're stealing oxygen as well. And just think about a boat trying to move through this, right? It's impacting recreation as well, boating, swimming, the flow of water, right? This is even more obvious if you look at the side of a dock. I mean, look at this. Even fisher folk who always think like, you know, oh, I'm going to fish in and around weeds. A fish cannot swim very effectively through this. I mean, this is so thick in this area here that fish do not enjoy this type of environment. There's not much room to move. It's soupy. It's low nutrients, low oxygen levels. So this is not a good environment for even the fish that we like to fish for as well. And one of the main things that I'm going to be stressing throughout this webinar is that if you put your boat into a system like this, Hydrilla can hang on to trailers and your boat. So clean, drain, dry. And I'll go over the steps to that in a little bit. Just for the record, it's called Hydrilla because it's named after Hydra, which is the 9-headed serpent of Greek myth. And that's because it can essentially grow an entirely new plant from just a tiny stem fragment. I thought I'd point that out as a cool like little fact. So what are we doing about it? Well, here in the lower Hudson PRISM region, they are using herbicide treatments. So really low doses of fluoridone in the Croton River over a 90 to 100 day interval. And there's a couple of different areas in which they've applied that treatment over the years. So they started that treatment in 2017 here, and it's been really effective at stemming Hydrilla. So this is just one example in the lower river of the Croton as to how it's gone from like 65% abundance and totally taking over that area to almost nothing in certain parts of the river. So it's been a very effective method of treatment. And not only that, so it's one thing to get rid of invasive species, right? But what we're also focusing on wild celeriae restoration, and this is a native aquatic plant that they're now restoring to areas of like that, and this is just on the brink of the Hudson River, and kind of bringing back that native habitat that a lot of our fish and our native food webs thrive upon, right? So it provides habitat and food for fish and vertebrates in our region. I also wanted to kind of show you guys a cool, fun, most wanted video as a promo from the Finger Lakes PRISM, because remember, this is the species that they nominated. And there's an amazing story map in the Finger Lakes that tells about the history of Hydrilla treatment in the Finger Lakes region. You can Google, it's called Battling the Hydra. It's treatment of Hydrilla in Cayuga Lake. I will also put it in the chat in a second. This is what that page looks like. So I'm going to enter it into the chat now so you guys can access that later if you want. There you go. And just to show you their little promo video, which I think is kind of cool. It's going to play for you in a second. Someone talked about lookalikes or asked about lookalikes. And again, it's got the toothy edges to its whorls. And usually the whorls are, you know, about 5 blades that are coming off of that. Very distinctive. So the Finger Lakes has a whole program now for photographing and then sending it to FLX, the Finger Lakes, plantid@gmail.com. And of course, there's other reporting that you can do, even if you're not in the Finger Lakes region. Of course, you can use iNaturalist or IMAP Invasives, which is the official New York State database for reporting invasive species. And there you go. To learn more, you know, here is your, and again, we are recording this webinar, so you can go back and see all of these great, wonderful links to all of these resources. But that's Hydrilla for you. So I think we are ready to move on to our next one, if I'm not mistaken. Yes, we are. So that was Hydrilla. That was Finger Lakes PRISM. And again, we have it here in the lower Hudson PRISM area as well. Okay, So SLELO, or St. Lawrence or Eastern Lake Ontario PRISM, is focusing on jumping worms. And I must say that jumping worms are found throughout the state now, and people now are becoming more aware that what they thought were originally earthworms are in fact these jumping worms. So we're going to go over that right now as to how to know the difference and what you can do about reporting and how to deal with it, right? So jumping worms are native to Japan and Korea. They arrived in Wisconsin in 2013, and probably from people selling mulch, soil, plants. That's how they're moving around. So the worms and the worm eggs, you can see them right here. They're really, really small, and they look like dirt. So if you're transporting plants or at plant sales and stuff, this is a message I really want to hit home, is to be very careful about transporting soil from one area to another. These worms tend to live just under the surface of grasses and forest under and the forest floor, right? So just under the surface. Earthworms, European night crawlers, tend to go a little bit deeper, and we're going to talk about some of those differences in a second. So, but let's take a step back for a second. New York's worms. I did not know this till I started getting into this field, but did you realize that there are no worms that are really native to New York due to glacial activity? Almost every earthworm in the US is non-native. So native earthworms all but disappeared about 10,000 years ago when the glaciers retreated. You can see this map here. There's where that glacial line is. So anything north of that kind of scoured it out north of Pennsylvania. New earthworms entered the US through settlers in about the 1600s, probably bringing over plants and stuff. And you know, we were taught since like grade school that worms in general are good for the environment, and a lot of times they can be, especially in compact soils or gardens where you're trying to get you know, some mix of nutrients. So you know, we know that they make holes in the soil, right? That's what we've been taught. But when they're at high abundance, especially along pristine areas of forest, and particularly with these jumping worms in really high densities that feed just beneath the forest soil layer, like right at the surface, right? That when you get in high numbers, it stimulates leaching of water and nutrients through the soil, and they turn over that duff layer, or that initial layer of the forest. You know, when you're stepping on a spongy duff layer of a forest in a pristine area, right? They eat that stuff so quickly that it can't be absorbed by the plants, and it just gets lost down, and the plants can't take it up fast enough. So the turning over of the soil can be too fast, especially with these jumping worms. So how do you know that you're looking at a jumping worm versus like a European nightcrawler, which again are kind of considered non-native, even though they've been here for a while? So first of all, jumping worms have their bodies a little sleeker and drier versus a nightcrawler is kind of slimier, a little floppier, right? The other thing is they have a very light colored clitellum. That's where they store their eggs and the gametes and stuff. And in a jumping worm, it's usually whitish and kind of versus like an earthworm, it's more raised up and a little bit more pinkish. So if I felt a jumping worm, this would be flush with its body versus a nightcrawler would be a little bit raised. But honestly, the biggest difference between these is their behavior. If you haven't seen this before, just wait till you see why they are called jumping worms. So this is something I took from Wisconsin DNR as to what they look like and how they move around. I will tell you that night crawlers do not do this. They move in a snake-like fashion. This is not sped up. This is in real time. So there is just this movement of like the serpentine movement and fast and jumping just does not happen with night crawlers. Our friends at SLELO PRISM also took a video along a plot that they were surveying for and kind of stimulated that worm to move. And you can see that jumping all the way from like this area of the plot to like almost like a foot away just by touching it. So watch it jump.
There it is.
Jumped almost a foot over to another plot. Earthworms will not do this. Yeah, they may try to squiggle out of your way, but they are not going to jump and move in that sort of like snake, like really, really fast fashion. So that's probably the best way to tell them apart in addition to those body parts. All right, on to the next slide. The other thing is that we need to be mindful of are their castings. It's another signal to or symbol or yeah, I guess a signal for them that they are in that environment. So that frass or that poop that they leave behind after they're done feeding on that right under that surface of your grass or forest floor, they leave behind these castings. Like when you run like water through your coffee filter and you're left with the coffee grounds afterwards, it kind of looks like that, or maybe like ground beef that you've just like put on your stove or whatever. This is what the cocoons are, and they're called cocoons. They contain the eggs. So these worms are parthenogenic, and what that means is that they have the ability to self-fertilize. They do not require mates for the next generation. So these little cocoons that the females can lay, they don't even need sexual reproduction. And they overwinter, these little cocoons overwinter in the soil to hatch in the spring. So a lot of different mechanisms for their spread. And that's why we got to be really careful about how we're moving plants and soil. So you can spread them as live worms or cocoons. You even got to be careful, like fisher folk going out, they think they got a night crawler or they think they got a worm, and then they're like dumping their bait out near waterways and in forests, like near the waterway. So that's another way it can move. Even those little cocoons can, soil on tires, hiking boots. So be really, really mindful of like just looking for the presence of this. And if you were in an area where you saw that sort of like that coffee filter soil, or you saw the worms in person, just be very, very careful of, using boot brushes, cleaning your clothes before you go to a new environment. I know that here in the Lower Hudson PRISM region, they have really changed how they do run their plant sales. So they're now sterilizing a lot of their soil before they transfer it. So they're growing it in greenhouses, sterilizing the soil. It used to be that they just took them from gardens of the master gardeners and then put them in their plant sales. So just something to be mindful of moving forward with some of your plant sales in the future. All right. In terms of control options, there's not a end all be all here. There's few pesticides that are available for worms. I had grubs in my yard, and I put down like a anti-grub chemical that is on my yard. And it actually killed these jumping worms, and I was able to pick them off. You can also dissolve a 1/4 cup of ground mustard seed in a gallon of water, and that'll like irritate the worms and allow them to come to the surface of your grass or your property, and you can get rid of them that way. And again, just kind of like solarizing your soil to eliminate the buildup of those worms and the wheat and kill worms and weed seeds that may be lingering in that soil as well. Okay, moving on. We are now on to the Adirondack Park, or APIP focal species, and that is the emerald ash borer. So this is, if you have it in your region, you know about it. So this is devastated a lot of the ashes throughout New York State. And this is what the actual adult bug looks like. It's an invasive beetle. It kills ash trees usually within 1 to 5 years and severely weakens them. They lose about 80% of their structural integrity even after, once the tree has died. And usually it's dead within 3 to 5 years of an infestation of these emerald ash borer. They've got, they're pretty small, but with metallic green wings, a copper abdomen. And guys, the main thing to be thinking about, especially if you go camping and you enjoy camping, its main mechanism of spread is in nursery stock and moving that way, but also spread being firewood. And we're going to talk about that at the end of this session here. But first, you got to know what an ash looks like. And actually, ashes are not too hard to identify because they have what are called compound leaves, and their leaflets come out opposite of one another. So you see how this leaf comes off that stem, or that leaflet comes off the stem, and there's another one behind it. Its branching and budding is also opposite, meaning that, each of its branches kind of come out opposite along that main stem. And even its cute little buds kind of come off opposite of one another. Its seeds look like long and paddle shaped. And even if you couldn't even see the leaves, or it was hard to see the seeds or not the right time of season, you can identify it by its bark. Its bark is pretty distinctive. It has diamond-shaped ridges on it, sort of similar to a walnut tree. So that's just another way to identify it in the field. Now, getting back to the bug itself. So the emerald ash borer deposits its eggs individually on ash trees, and it's usually between the outer layers of bark and in cracks and crevices of the trunk and the branches. So the female ash borers lay between 40 to 70 eggs, kind of within those cracks of a bark of an ash tree. Then within 7 to 10 days of that egg laying, and those eggs are laid in June to August, within 7 to 10 days, you're going to see this little larva, this white larva. And the way that it does it is it's insidious. It kind of like crawls in the living tissue of that tree. So it feeds on the phloem of the tree. So that's the pipeline through which food is passed from the leaves to the rest of the tree. And it also feeds on the cambium, which is the growing part of the trunk. So essentially it's feeding on the pipeline that the ash uses to get nutrients all throughout its, the plant itself. So it ends up killing the tree eventually. And then eventually those larvae will emerge, they turn into adults, and then they chew a telltale D-shaped exit hole in the bark. And then the adults can then fly, and then they seek out new trees and begin the process again. Well, I'll show you like actual pictures of what this looks like. So again, the larva is crawling in the living tissues that the kind of the lifeblood of that tree, and it leaves behind these S-shaped galleries. And the way that I remember that is like it's like an art gallery. I mean, it really is kind of really neat to look at, but it's sort of like a death art, right? I mean, it's killing the tree. You peel back the bark, and this is what you see. That's the telltale sign of an emerald ash borer infestation. That leads to canopy dieback and canopy thinning. So after a while, it kind of loses its nutrients and ability to grow. So a lot of canopies then die back. You might also see ash trees that look like this. And you're like, what is causing that? Like something like, what happened to the bark here? And that's actually due to woodpeckers that know that the bug is in there, and they'll start peeling back the bark and causing this like blonding or strip bark appearance, and these big deeper holes due to woodpecker damage as well that are going in there to feed on this like dying tree. Another kind of stress response that the tree has are these epicormic shoots. And that is just like, oh my God, I'm like, the tree is stressed out and it's trying to just shoot out new shoots from its base. And those are called epicormic shoots, and just another sign that this tree is under dramatic stress from this beetle. You'll also see bark splitting. And again, if you peeled back that bark, you would see those S-shaped markings that look like this, and those serpentine galleries formed by that larval feeding. That is that D-shaped exit hole that you will see on the bark as the adults emerge and then begin to fly away from that. So you can also be on the lookout for that in ash trees as well. And because this was the focal species for Adirondack Park, I did want to say that unfortunately, due to firewood movement, they now have 11 new confirmed locations in and around the Adirondack Park. So you can see that those are up here, and then 4 within the park itself. And there is major concern. They're doing everything they can to keep it out of the Adirondacks. And a lot of the messaging is about do not move firewood from one location of New York State to another. New York State laws and regulations say no more than 50 miles, but within the Adirondacks, they're saying just buy locally sourced firewood or and just leave it at that. That way, there's no way that you can introduce it through firewood if you're buying it locally from the Adirondack Park. So in terms of how you can help, again, buy it where you burn it, right? So make sure you're buying locally sourced firewood, or that it has the label on it, New York State heat approved, right? Do not do the heat treatment yourself. There are certain stacks that you can buy that have a specific label on it, but really recommend buying local firewood. There's also a lot of different citizen science opportunities, right? So you can use IMAP invasives, and you can see the website here. There's a lot of great webinars and how to use this citizen science tool. And this is, again, the official reporting and mapping database for New York State. There's also monitoring and managing ash, which is a type of a citizen science project that looks at lingering ash trees. So these are ash trees that are found in areas that are devastated by the emerald ash borer, that, there's something about their genetics that they're able to withstand it. And they're looking for what about it, so that we can begin to repopulate and rehabilitate a lot of these forests that were devastated by emerald ash borer. And if you're interested in what Adirondack Park is doing for biocontrol release sites, you can see this email address to get in contact with them. All right, moving on to Capital Mohawk PRISM. So Capital Mohawk is going to be focusing on European frogbit. Now, European frogbit is a free-floating aquatic invasive plant, and again, just native to Europe and Northern Asia, and introduction through ornamental escape. It was first reported in New York State in 1974, and you can see that it's pretty centered around the Great Lakes area and in the Northeast. So its primary area where it was first introduced was in Lake Superior, and it's kind of made its way east across. In terms of its ecology and habitat, it really prefers slow-moving marshes, rivers, lake shorelines. It's really tolerant of high calcium levels. So although it floats, it's considered to be a floating plant. It does not have very deep root system. It doesn't shoot down its roots like to the sediments itself. And so they're usually found in shallower waters of less than 2.5 feet. But again, forming these dense mats that block sunlight from a lot of the native submerged species that we're trying to protect. And the reason why it forms these dense mats is that I'm throwing a lot of plant terms at you guys here, but it reproduces through stolons, which are horizontal stems. You can see it here that grow away from the parent and then form their own roots and stems to form new plants. So it's like a whole bunch of plants holding hands together. So imagine this like dense mat moving across the surface, and just with these really, really shallow roots here across in all different angles and areas, and creating these dense mats. They also reproduce through turions, which we talked about before, which are, go dormant and overwinter. So this is its main mechanism of spread, and it's not really through seed. It's like through these colonizing characteristics that it has. In terms of its ecological impacts, you can see this dense mat. I mean, early spring growth, outgrowing competing native plants, low dissolved oxygen, and it just dominates shallow waters, impeding water flow, right? So in terms of ID, what should you be on the lookout for? Well, the leaves tend to be pretty small, actually, just a couple of inches, like not very big at all. Like this is being held in a hand here. They're kind of like heart-shaped and rounded. They have a smooth margin on the end. You can see its 3 white petal flower. But the main thing I want to point out about its ID is that it's petiolate. In other words, it's got multiple leaves that are attached to a single stem. You can see those shallow roots. I wanted to show you, we have it here in the Lower Hudson Prism, and my colleague just the other day went out and took a view through a kayak of what she was finding. That's European frogbit right there. You can see it kind of looks lily pad-ish, but again, it's more like they're very, very small, just a few inches, and it's got like more of a heart shape to it. And again, she's going to show you underwater. Unlike a lot of the lilies, this has very, very, you see that? Just those roots are just a little bit below the surface, right? Versus the lilies will have much, much deeper roots. And all along this area here, just creating these dense mats, very, very small leaves, and just completely covering this area that she's kayaking here. So this is what it's going to look like at the surface. And I think you'll even see Lindsay, my colleague, exclaim here, just covering this area.
This is great. Just getting like all this.
So that is European frogbits. So hopefully you can be on the lookout for that and help us map it in our region. Okay, moving on to the next invasive species. Oh, in terms of management and control, yeah. Again, clean, drain, dry. Clean off visible aquatic plants and mud from equipment before you move from one system to the next. Drain, watercraft bilge, live wells, motor, other water. containing devices before leaving the waterway, right? And getting into a new system, and dry everything for at least 5 days, or with a towel if you don't have the 5 days. So wipe it down after you're able to clean and drain. That is the recommended way of control. All right, moving on to CRISP. So CRISP is in the Catskill region, and they decided to focus on jetbead. It's also called black jetbead. And as you can see here, it's a multi-stemmed deciduous shrub that can grow to be about 6 feet high. You can see its growth pattern here. It's got these very kind of showy, conspicuous white flowers that shows in late April. It's probably done blooming at this point in our region. You might see some lingering flowers there, but you know, again, it was introduced in the late 80s from native range in China, Japan, and Korea for, guess what? Its ornamental value. And it's its flowers and even a kind of its leaf that gives it that ornamental pop. And it still has, you know, prized ornamental value, but it's escaped cultivation. It's now widespread in the northeastern and midwestern United States, and found in 23 different states. It can establish itself in a lot of different areas. So roadside banks, forest edges, understory. And unfortunately, you know, it can grow in both full shade, but prefers full sun. And that's a lot of the different areas where it occupies a position in the forest that would normally support native understory shrubs, like maple leaf viburnum, low bush blueberry, right? Once it's established, it shades out those native plants in the ground layer and inhibits native tree regeneration. And again, like a lot of invasive species, right? Adaptable, various soil pHs, poor soil conditions, shade, sun, and it spreads by sprawling. It can reproduce by what are called basal suckers. So again, coming out from the roots and spreading horizontally from there and sprouting new plants. And its vectors, once established, include small mammals and birds, but a lot of the movement is through the horticultural trade. So I'm going to show you some of its ID features in this ID video. And again, if you are an ISF or invasive strike force surveyor with us and you do the intermediate training, we find it here in the lower Hudson PRISM region. And this is on our list and what we are actively trying to map in our region, as well as the Catskills.
So first of all, as you can see, are its berries. So the berries grow in a very unique fashion. You can see that there's 4 dark berries that grow almost perpendicular to one another. And these usually come out in the late fall, but these have persisted. And that's part of the reason why it's troublesome as an invasive, is that you've got these very persistent berries, birds will eat them and then widely disperse them.
So this is an important characteristic. So the berries are persistent, and it's very, very neat. It's like a cross, like almost like a plus sign of these black berries. So you may see that even persisting now.
And then right next to it, you can see its flower. So it starts flowering in spring. So again, late April or May that you'll start to see that. And it's just, you know, this white flower. They don't grow in clusters. So you'll just see this one prominent flower in between a couple of leaves, and the leaves grow opposite of one another, as you can see. So like they come out right next to one another. The other key distinguishing feature are its leaves. So if you take a look, see how the leaves are very toothed? All right. So, and then they're very deeply veined. That's another key characteristic. And if you look at the top of this leaf, you'll see that it's got like sort of it's very elongated towards the top, if my camera comes into focus here. So you see that like sort of elongated tip. That's another key feature, as well as the toothed margins. It's not quite as deeply toothed.
So yeah, I mean, if you felt this leaf, it feels like kind of rough, and it comes to that point and a lot of like really deep teeth. And it's got like, I don't know how else to say it, like really, really deep, noticeable veins that are coming off of it, in addition to what we just highlighted in terms of, you know, it's white flowers, which you may not see this time of year, but again, in the early spring, you will. It's oppositely branched leaves and those ribbed veins that are elongated at the tips. And if you see these berries, there's not much else that's like it in the area. They turn dark red to black, usually in late fall, but again, they can persist throughout the winter, and you might see them in the spring as well. All right, Guess what, guys? We're moving on to my PRISM, where I am located in the Lower Hudson PRISM area. And we are going to be focusing on water chestnut. And you might be asking yourself, Brent, isn't this a common invasive species? Many of you guys may have it in your areas, and you'll know that typically where water chestnut is, it can really be obvious and take over an area. So this is a species that was native to Europe, Asia, and Africa, and is one of the first ones to make it over here to New York State. It was first reported in 1884, introduced again through the ornamental trade, and it's pretty much like sequestered to the Northeast. Now, again, a lot of you may consider this to be common, but here in the Lower Hudson PRISM region, it sort of is very common in the Hudson River, but it's much less common in inland water systems. So this is something that I want to point out, that even though it might be widespread in certain local ecosystems, we are really trying to prevent it from getting into inland water bodies like lakes and areas that are uninfested, because usually once this takes a hold, it can be very difficult to get rid of. So young local infestations still have the potential for eradication, and that's why we're focusing on it today. And I'm going to turn this over to my colleague, Lindsay Yoder, who is the Aquatic Invasive Strike Force coordinator in our region. And she created, and her team last year created this amazing video on managing invasive water chestnut that I wanted to share with you guys. It's really educational and a good educational video for you. So we're going to play that for you right now. And you'll learn about not only its ID characteristics, but also how we're managing it here in the Lower Hudson PRISM region.
Oh, hi. We didn't see you there. I'm Kate. And I'm Eleanor. And we're with the Aquatic Invasive Strike Force. Today, we're removing the invasive water chestnut.
So it's a floating plant.
This is the invasive water chestnut. It forms dense mats on the water's surface and prevents sunlight from penetrating through the water column, which stops native plant growth. Other impacts of water chestnut include blocking pathways for boaters and fisherfolk, as well as depleting the dissolved oxygen within the water. This is because when the plant dies off, the bacteria that decompose it consume most of the oxygen in the water. This can also lead to harmful algal blooms. Timing is critical for water chestnut removal. The ideal removal period is from mid-June to late July, and if needed, early August. The reason for this timing is because around August, the seed pods begin to drop, and each seed pod can produce up to 20 plants, and each plant can produce up to 20 seeds.
In order to effectively remove water chestnut, we have to remove the whole plant, including the roots. And so we start with the rosette and making sure.
And you can see the roots there. And I'm going to point out what that nutlet is. And I'm sure if you've been in a water chestnut infestation area, you'll see it. This is dark, like really sharp nutlets that turn like kind of a darker color, especially as they're washing up.
Try not to break off the roots. Carefully pull up the entire plant. As Kate mentioned, each seed can turn into multiple plants. So we want to make sure to get the roots so we can get the residual seeds on the bottom.
And there's that nutlet there and the roots that they're pulling out.
When disposing of the water chestnut, it's very important to do it properly, whether it's throwing them in trash bags or placing them 100 feet away from the water to decompose naturally. Thanks for tuning in, and we hope to see you protecting New York's waters, one water chestnut removal at a time.
All right, so that's water chestnut. And again, there's lots of ways that you can follow us on Instagram and Facebook, and the amazing work that our aquatics team is doing on water chestnut. They have a lot of community volunteer events on this, and that's usually in July and August time that they start doing that. So, and we've had some great great success that I want to share with you in some of these removals. So let me tell you a little bit of a success story of our team here in the Lower Hudson PRISM region. So this is a small lake in Putnam County Veterans Memorial Park. It gets a lot of like recreational use there, and water chestnuts were first spotted in 2018 by those summer residents. So lots of swimming, boating, and recreation, sunbathing, that sort of thing in this lake. So in 2019, our aquatics team went out there and removed 21,000 plants from this area. And this was all done with volunteer help. It's amazing what you can accomplish in a short amount of time. But, you know, it's one thing to just be like, oh, okay, well, doesn't this just come back through reproduction? Well, they went out the next year and they only had to remove 2,850 plants were removed from that region. And let's look at a before and after picture of that. So this is a huge success story. It does really make a difference, like you putting in the time and resources to do this. And, you know, we're hopeful that even though water chestnut seeds can last over a decade, we're hopeful that through this rapid response, that will assist in the successful eradication of this within a few years. So that's the goal and the hope. Just wanted to celebrate that success story with you. All right, and rounding the final turn here, guys, we're now going to be focusing on our last species. Long Island, or Lisma, the focal species, is on hardy kiwi, and you can see some photos here. Again, I'm going to turn this over to one of my colleagues, who is Joyce Tomaselli from our Cornell Cooperative Extension in Dutchess County. And even though she's not here live, as Lindsay wasn't here for the last video, I'm going to show you a video because she was able to locate hardy kiwi as a naturalized hardy kiwi and tell you a little bit about it. So I'm going to turn it over to Joyce. If I can find it, lots of tabs open here. Give me a second. Oh, it's on my QuickTime. That's where it is. All right, here we go.
Hello, my name is Joyce Tomaselli from Cornell Cooperative Extension, Dutchess County. Here to talk with you today about the vine called hardy kiwi. Hardy kiwi was introduced, as many of our invasives were, back in the late 1870s, early 1880s, as an ornamental vine. It has nice glossy leaves, it's vigorous, it loves to climb and grow. Actually, hardy kiwi can grow as much as 20 feet in a year. It is also the darling of some small farmers today because it has delicious fruit. Let's look at a couple of details here on hardy kiwi. It is a large vine. It can grow 20 to 30 feet or as tall as the structure it can find to grow upon. It is deciduous. You do see the woody vine persisting during the winter. It branches in an alternate style as opposed to opposite. The leaves are finely serrate. They're zigzagged on the side. And what's going to be key as we look at the identification is that the petiole, which is the bit of the stem here from the end of the leaf down to the...
So the petiole is essentially like the leaf stalk. So you can see the blade, the leaf blade here, and then kind of going down to the stem. So that's really important ID feature, is that it's red.
Bud, that petiole is red. And that's going to be key for you to understand hardy kiwi compared to bittersweet.
The video transitions really quickly. So she's going to talk about the fruits of this, and they are edible. And that's why it was planted, I think, because of its the edible kiwi, and thought it would be a delicacy kind of thing. But they're really grape size, and they're green. They're actually very small, not like the kiwis you might buy in the store, but they are related.
It flowers late summer. The fruits are about the size of a green grape. This is hardy kiwi, Actinidia arguta. It is a perennial woody vine, and like bittersweet, its leaves are in an alternate arrangement. They're simple, they're lobed, they're serrated, and the ones on...
By serrated, again, you see the teeth on the end of that? That's what she means by serrated.
Newer growth are often more pointy than the ones on the older growth. This plant has some flower buds. They'll open up in another couple of weeks. Right now we're at the end of May. This plant can get really huge. The key thing with hardy kiwi is that the petioles, the stems,
are red. This plant, just like bittersweet, is a heavy twiner. It twines around itself.
So as you zoom in here, and I'll show you a bittersweet in a second, just for sake of comparison, but you can see that it's wrapping and corkscrewing around itself. So it's a twisty vine, even though it kind of looks like it's growing like a shrub, it really is a vine. And so I wanted to just end with a few things here, or kind of rounding the end here. But first of all, how it differs from Oriental bittersweet. So Oriental bittersweet is a common invasive all throughout New York State. But you can see that the fruits of Oriental bittersweet are actually quite different. They come out like, like late summer, like fall time. And again, it was because, a lot of people like to put this in their pergolas and stuff. It kind of persists throughout the winter. But you can see that those berries are orangish or reddish versus the kind of like the green grape-like of hardy kiwi. And again, bittersweet can corkscrew and girdle trees very similar to hardy kiwi. But the main difference is that, remember that even though bittersweet can have these pointier leaves that are toothed on the margins, especially when they're young, remember, it's the red petiole. Oriental bittersweet has a green, or it's like woody, right? Versus hardy kiwi, it's going to be red. And that is how to tell the difference. In terms of hardy kiwi management, again, this is something that Long Island is focused on, but we are actively managing here in the Lower Hudson PRISM. We are doing a lot of cutting and removing plus herbicide. So just this past year in an infestation area here, our seasonal crew members removed over 211 individual plants all along this fence and this other natural area. But just be careful if you are removing it, that they have underground runners that are abundant and rapidly regenerate. Are you noticing a theme on that? This underground system that's going on that's very difficult to remove. They use 3% glyphosate for removal. Usually cut stump is most effective. But just, if you are just doing manual, the stem nodes in contact with the soil can resprout. So all of these things make it difficult, but not impossible, right? If you get the right help. So with that, I just wanted to say thank you, and also just put out there that it's amazing to not only be working with the amazing volunteers that I'm blessed to work with, like yourselves, that are here and care and want to do and take the next steps and learn and be educated and work with us. But also, we are part of this wonderful team of this PRISM network that is connected. Like, I got to thank all of them for their help with some of the slides. And you can see just across the state how we're really trying to work to improve the conditions of our natural areas and keep them open and accessible to the public for us to enjoy, and also bring and restore resiliency and that sort of like that natural beauty for our ecosystems that we love. So with that, I know that we're out of time, so I'm going to stop the recording here, but I'm happy to stay on and take questions for you. I can see that the chat box did have questions as we went on, but thank you again for being such a great audience, and hope you have a great night. And again, I'm here to take questions if you got them. Thanks, guys.